Hi medicinites. In this video we will be seeing about what is the introduction to physiology, and we will be telling about how to study physiology, and we will be seeing mainly in this video about the cell structure and the different structures about in details, and we looking about their functions individually, and we will be going through different structures in the cells. So guys, what is physiology? Physiology is a study of functions of human body. So what is the best book to prefer to study for physiology? Physiology is generally used to study among the world and is universally accepted books, which is the one among is that of the Guyton and all. This book is very universally accepted and is studied over different parts of the world. So what is the best way that can be studied to physiology to remember things for a long time? To remember things for a long time, one can make mnemonics and to study their concept using most diagrammatic way, so that they don't forget things in a short period of time, and will help to recall them within a simple, very short span of time. Hi guys, let's start with a tour of our cell. The first structure we are going to see is about this is the big mama of the cell, and this is the located in the center of the cell, and it is also known as the brain of the cell. You know what is the name of this structure is known as? Hi guys, this structure is known as the nucleus. Nucleus contains different parts. The first part we are going to see is about the outer envelope. The outer envelope contains many ribosomes attached to it. The next structure is the inner envelope. Inner envelope, it contains a protein known as a lamin protein. These lamin proteins are involved in the attachment of chromatin to its together with them. Any mutations of the lamin proteins, they cause a disease known as progeria. These blue structures, these are known as the chromatin. They are of two types. These are of heterochromatin and euchromatin. The heterochromatin is transcriptionally inactive and they are very densely packed, whereas the euchromatin is transcriptionally active and they are very loosely packed. The location of euchromatin is usually in the center of the nucleus, whereas the location of heterochromatin is usually towards the inner envelope, that is the periphery of the nucleus. Contains these structures here. These are known as the heterochromatin, and these towards the center, these are the euchromatin. And these red proteins, these are here. These play a very important role in the nucleus. These are known as the nucleopore. These structures, they contain different types of transport proteins in them. These are contains many transporters which are involved in the transport of substance between the nucleoplasm and the cytoplasm of the cell. They transport different types of proteins, ribosomes, and the RNAs and the different ions through these nucleopores. So, transport is mainly involved in transport. This red structure here, this is known as the nucleolus. It mainly contains the rRNA, which is the ribosomal RNA, which is involved in the formation of ribosomes. These ribosomes, which are formed from the rRNA, they get attached to the nuclear envelope, which is the outer nuclear envelope mostly, and the different envelopes of different membranes of the cell organelles usually. And these chromatins, they are usually made up of DNA, and they also contain histone proteins. Nucleolus, RNA contain rRNA, chromatin, DNA plus histone proteins. From the nucleus, from the nucleolus, these mRNA, they get transferred towards the cytoplasm through these nucleopores. Maybe the mRNA, different types of RNA, mRNA, rRNA, and different types of RNA are transferred to the cytoplasm through these nuclear pores, and they move towards a different cell organelles in their further movement towards the cell. There are different events that occurs in the nucleus. These are the DNA replication, DNA replication is the process by which a DNA produces a new DNA from a former DNA, and that is the second one is the transcription. This is the process by which a DNA produces a new RNA. And the third one, then we will write the third one here to lack of space there. Third one is the translation. This is the process by which a RNA produces a protein. The next structure we are going to look is the endoplasmic reticulum. This is of two types. This is a smooth endoplasmic reticulum and this is a rough endoplasmic reticulum. The major difference between the smooth and the rough endoplasmic reticulum is the presence of ribosomes on the rough endoplasmic reticulum that makes the surface to look rougher. These ribosomes, these are attached to the outer surface of the endoplasmic reticulum, which is the filaments of the endoplasmic reticulum, making it appear to be rougher in appearance. The main function, it is that the RNA from the nucleus, they gets attached to the ribosomes in the endoplasmic reticulum and is involved in the synthesis of protein synthesis. We see a protein here. These proteins are synthesized and these proteins are get folded in the endoplasmic reticulum. What's protein folding? This protein folding is necessary for the functioning of the protein. These proteins are then is involved in the glycosylation. Glycosylation, it is the process by which the sugar residues are added to the protein. This is a glycosylation. The type of glycosylation that occurs in the rough endoplasmic reticulum is the N type of glycosylation, which is the addition of sugar residues to the nitrogen of the protein. And these are mainly involved in that of the protein synthesis. These proteins which are synthesized are made as a bud from this filament of the endoplasmic reticulum. These proteins are then released into the cytoplasm, which are then transferred to the Golgi apparatus. The next structure here, it is a smooth endoplasmic reticulum. They contain no ribosomes. These are mainly involved in the lipid synthesis. The lipid synthesis are of cholesterol. Cholesterol is involved in many steroid hormones, testosterone, aldosterone, steroid hormones. These are all involved in the phospholipid synthesis. They are also involved in the fatty acid synthesis, fatty glycogen synthesis. Fatty acid synthesis. They contain a substance known as CYP450. The CYP450 is found abundant in the liver. This is known as the cytochrome P450. This is involved in the detoxification of certain drugs, alcohol. This is known as the detoxification of substances, involved in the biodetoxification of substances. Detoxification, xenobiotic. The breakdown of certain substances. These smooth endoplasmic reticulum. They contain certain enzymes which are involved in the formation of glucose from glycogen. In cytoplasm, we have glycogen, which is a polymer of glucose. To produce glucose, it needs certain enzymes which is produced in the smooth endoplasmic reticulum to act upon them, because the glycogen converts into the glucose 6-phosphate, which then converts into glucose. The glucose 6-phosphate is acted upon by the enzyme that is produced from the smooth endoplasmic reticulum, which takes away the phosphate from the glucose 6-phosphate and is later then converted into the glucose. And these substances which are produced from the smooth endoplasmic reticulum are then made as bud from the smooth endoplasmic reticulum, and they detach from this smooth endoplasmic reticulum, and these further they travel to the Golgi apparatus along with that of the vesicles that come from the rough endoplasmic reticulum. These both substances move towards the Golgi apparatus. It contains a lot of calcium ions in this. These calcium ions are involved in the muscle contraction. The smooth endoplasmic reticulum in the smooth muscles are known as the sarcoplasmic reticulum. Sarcoplasmic reticulum. The sarcoplasmic reticulum contains a lot of transport of calcium, and these are involved in the muscle contraction. The next structure we are going to look about is the Golgi apparatus. The vesicles from the rough endoplasmic reticulum and the vesicles that comes from the smooth endoplasmic reticulum, these enter into the cis phase of the Golgi apparatus. Anatomically, the Golgi apparatus consists of two phases. This is the cis phase. The vesicles that enter into the Golgi apparatus, it is the cis phase, and the substance that leave the Golgi apparatus is the trans phase. In the Golgi apparatus, these vesicles undergo certain modifications. These modifications involve glycosylations. This is the glycosylation, and it also includes the phosphorylation. Glycosylation here it involves of two types, that is the N-type glycosylation and the O-type glycosylation. Whereas in the endoplasmic reticulum, it is only the rough endoplasmic reticulum, it is only the N-type glycosylation happens there. In the O-type glycosylation, the proteins are added with the sugar residues with that of the oxygen, whereas in the N-type it is with the nitrogen. These are two types. This is the N-type and the O-type. And these vesicles which pass through the Golgi apparatus towards the trans phase, they form a embulging of this Golgi apparatus as a bud, and they leave the Golgi apparatus as a vesicle, and these vesicles contain protein. These proteins may be proteins of the lysosome. They may be a membrane protein, and these proteins can be those which are excreted out of the cell. Next structure we are going to see is the cell membrane. Cell membrane consists of the phospholipid bilayer, and it also contains the fatty acids. The cell membrane, it acts as a barrier which is involved in the allowing the entry of substance inside the cell. These are selectively permeable, which do not allow almost every substance to enter into the cell. They selectively allow substance to enter into the cell. These contain some carbohydrates. When there is a high amount of carbohydrates in the cell membrane, and there is a very less fluidity among the phospholipids, as these phospholipids do not move very easily inside the cell membrane when there is high amount of carbohydrates, whereas when there is low amount of carbohydrates, the fluidity increases in the cell membrane. It contains different type of proteins. These proteins can be of different types. These are peripheral proteins and integral proteins. These peripheral proteins are very loosely bound to the cell membrane, whereas integral proteins, they are very tightly attached to the cell membrane, and very much so much of energy is needed to extract these integral proteins out of the cell. The next structure we are going to see is the lysosome. These are the lysosome. These are spherical organelles. These are spherical organelles. They contain many hydrolytic enzymes. These enzymes may be protease, lipase, nuclease. Protease are enzymes that break down the proteins, whereas lipase are enzymes that break down the lipids, and nuclease are those which break down the nucleic acids. These are involved in the breakdown of macromolecules. Macromolecule breakdown. Macromolecule breakdown. These digestive proteins, carbohydrates, and lipids, and they are also involved in the autophagy. Autophagy is the activity of the lysosomes which is done on the those organelles which loses their functional activity. For example, if these ribosomes, these are about to lose their functional activity, these lysosomes act on these ribosomes. These ribosomes are generally made up of RNA and proteins. So these lysosomes that act on them, which uses these nucleases and proteases to digest these ribosomes, and this process is known as the autophagy. Whereas the autolysis, it also performs a function known as autolysis. Autolysis is the function through which, when a cell is damaged, these lysosomes, they break open and they destroy the entire cell. This function is of the lysosome is known as the autolysis. The next structure we are going to see is the peroxisome. This peroxisome contains many enzymes. These are catalase, oxidase, and metabolic enzymes. The main function of the peroxisome is that they help in the removal of the free radicals. Free radicals, sometimes oxygen can be converted to superoxide, O2-, hydrogen peroxide, and these substances are very harmful to the cells. They attach to protein cell membrane and cause damage to them. These peroxisomes are involved in the conversion of the hydrogen peroxide to convert hydrogen peroxide to water and oxygen, which makes very harmful substances into less harmful substances. These are necessary substances for a cell, water and oxygen. They contains they are involved in the lipid synthesis and lipid and cholesterol synthesis. Catalase aids in the first step of the fatty acid synthesis. They break down the fatty acid, that is the fatty acid oxidation. Catalase aids in the fatty acid oxidation. They break the fatty acid into the acetyl CoA. Fatty acid oxidation, it is of two types. That is the alpha oxidation and the beta oxidation. Alpha oxidation and beta oxidation. Alpha oxidation, it occurs in the branched chain fatty acids, and beta oxidation occurs in the very long chain fatty acids. They break the substance to fatty acids to acetyl CoA. And these structures are also involved in the ethanol metabolism. These red structures here, these are known as ribosomes. They contain two subunits. This is the larger subunit and the smaller subunit. And a small subunit. The large subunit is of 60 S. The S stands for Swebig unit, and the smaller subunit is about 40 S, 60 S in eukaryotes. The main function of the ribosome is the protein synthesis. The protein that is synthesized by these ribosomes, they are of two types. These are the cytosolic protein and these are membrane bound proteins. These membrane bound proteins are those which forms the proteins in the lysosomes, and those in the cell membrane, and those the proteins that are exited out of the cell, whereas the cytosolic proteins are those present in the cytoplasm. They don't leave the cell. They are involved in the synthesis of many enzymes in the cytoplasm. This structure here is known as the mitochondria. It contains of double membrane. This is the outer membrane. The outer membrane is permeable, highly permeable outer membrane, and is very smooth, permeable, smooth. It contains the inner membrane. It contains a lot of infolding. These are known as cristae. Inner membrane. And this part here is known as the matrix. It contains the mitochondrial DNA and some highly involved in the metabolic reactions. This part is of the mitochondria involved highly in the metabolic reactions. This mitochondrial DNA is to be noted that it is from the maternal part. It is the maternal part of DNA that forms the mitochondrial DNA. This structure is mainly involved in these many metabolic reactions like Krebs cycle, heme synthesis, urea cycle, and glyconeogenesis, and ATP synthesis. The ATP synthesis in mitochondria occurs by two different pathways. These are substrate level oxidation and two different pathways. Substrate level oxidation and oxidative phosphorylation. The oxidative phosphorylation occurs in the inner membrane of the mitochondria through the electron transport chain. This mitochondria is known as the powerhouse of the cell, and the glycogenesis that occurs in the mitochondria involves the conversion of the fatty acids, amino acids into glucose. This is known as the glyconeogenesis. Occurs in the mitochondria. And urea cycle, it converts harmful ammonia into urea, whereas heme synthesis, it converts pigment known as the hemoglobin in the hemoglobin. It converts production of hemoglobin which is present in the muscles. These are known as the myoglobin, and it involves Krebs cycle. Occurs in this mitochondria. Cytoskeleton includes the microfilaments. This is the intermediate filaments, and this is the microtubules. This is microfilament. The microfilaments is involved in the microfilaments, which is mainly it is known as the actin. When actin is combined with myosin, it involves in the muscle contraction. This is involved in the changing the shape of the WBC and causing it to diabetes. Diabetes is known as the squeezing out of WBC from the bloodstream. It involves this WBC. It gets its shape change during change in shape, and it gets ejected out through these blood capillaries, through these gaps between them. These are known as fenestrated capillaries in them. They exit through these capillaries, and this is known as a diapedesis. And these are involved in the mainly provide support to the cells. And the next structure is known as the intermediate filaments. These intermediate filaments are involved in the process of anchoring. They anchor this cell to the extra cellular matrix, which contains many collagen fibers, different types of fibers. They anchor the cell to the different types of fibers in the extracellular matrix. They anchor one cell to another cell between the cells, and they anchor different types of cell organelles within the cells. The main purpose of this is anchorage. And these microfilaments, they are involved in the cell division. They form these when the cell is dividing, they form this they make a division between the two cells and are involved in the separation of the two cells. These two cells then become separated into two different cells, and this last one is in the tubules. It consists of two subunits. These are alpha, and this blue one is the beta subunit. These alpha and beta subunit together makes the microfilaments, and that makes microfilament. 13 microfilaments make the microtubules, and these microtubules are involved in the during the cell divisions. These microtubules attach to the kinetochore, and they separate the kinetochore into two sister chromatids during cell divisions. The main function of the microtubules is the involved in the cell division, and they also involved in the cell extensions like cilia and flagella. They form cilia and flagella. Cilia and flagella are involved in the movement of substance in the respiratory tract. In the reproductive tract, they are involved in the movement of substance, and ovum in the reproductive tract. These substance aid in the transportation of substances in the different parts of the body. Transportation. Hi my listeners, in this video we talked about the different cell structure and their functions. In the upcoming videos we will be seeing about the more explained video about the lysosomes and peroxisomes. I hope this video makes sense and you guys enjoyed it. Please press the like button and comment on the comment section, and press the subscribe button to get more videos from us.